Hello, students. We have an exciting module ahead of us, the animal microbiome. Many renowned conservationists believe that the best way to inspire change in our world is by educating others, by questioning them, asking what most amazes them about the world or about wildlife, and allowing them to discover for themselves how remarkable other life forms are. These remarkable animals touch our lives in many ways. Ecologically, they maintain balance in the habitats, ensuring the health of our planet. Culturally, they inspire myths and art and traditions around the globe, enriching our human experience. And on a personal level, they evoke a sense of wonder and connection. The study of biodiversity on our planet is, in a word, inspiring. So, to improve the health of our ecosystems and our planet, studying the microbiome offers a new means for conservation, and especially for bioremediation strategies and for new methodologies for protecting, especially vulnerable or endangered species. In this module, I'd like to explore several different fascinating aspects of species that I'm really interested in, including some of our personal favorites. And I use this in a way to focus on some overarching themes on what drives diversity. In this module, I really want you to be thinking about curiosities of your own as well. So we have implemented that into a small scaffolding assignment, which you will see on Canvas, microbiome curiosity, and you will be posting and sharing your own. Please make an original post. All of you are very familiar with this process by now. The post will be to a Canvas discussion board this time. You only have to post once, but be sure to look at the requirements. And then, as an extra credit opportunity, you're welcome to post after the deadline over 2 days to other your peers' curiosities for extra credit. Okay, so without further ado, let's go over some interesting studies, recent studies on the animal microbiome. We're going to dive into a few different species and what has different studies about these animals, including sea turtles, sharks, bees, birds, crocodiles, hyenas, and baboons. The bottom line up front is that the microbiome is diverse, adaptable, and influenced by many environmental factors. This is a theme that has consistently come through in your journal clubs as well as in the lectures and required readings. The microbiome interacts with the host in beneficial and harmful ways. This we know. But with respect to animals, it can help in digestion of food, with nutrients, stimulate the immune system. And I will provide some interesting new evidence where researchers at the University of Central Florida are asking the question on whether or not they can play a role in as like a magnetic compass of sorts, a microbial compass of sorts in navigation. And the microbiome also interacts with the host in terms of behavior. Animals and plants have very complex mechanisms for evading pathogens, harnessing their commensal microbes. This is another theme that we've seen before. And social interactions are probably the most important driver of composition, providing rich opportunities for microbial transmission. And this is known as the social microbiome. You'll see that there's a bit of required reading in this area this week. Uh, we're going to talk about various life forms, but marine life we'll find is influenced by salinity, temperature, water quality, and coral reefs play a really crucial role in maintaining the health and balance of this ecosystem. Um, that might be an area of interest to many of you that you want to present your curiosity on. In mammals, there's influences of environmental factors and social behavior, of course, reptiles as well. And we find that there are vast differences based on the body site, just as we do with humans. And in birds, roles in digestion, disease resistance, and migration patterns can actually play an active role in the composition of the microbiome. And in insects, there are really important aspects of immunity. So first, let's talk about a couple of curiosities with respect to the sea turtle microbiome. Here I've cited a study where the authors were interested in sampling the skin and the carapaces of 26 loggerhead sea turtles. And they chose these because they're a very important species in the Mediterranean marine environment. And the outer microbiome is very rarely studied as seen with other reptiles. They performed amplicon sequencing of 16S and 18S rRNA genes. They found that bacterial diversity and richness of the carapace was significantly, consistently higher. And this could be due to the fact that it's covered in scutes that could allow for better attachment of microbes and also subsequent colonization. This is interesting because in a bit we're going to talk about sharks as a contrast to that. Also, the skin of the neck in the flippers has to be quite flexible. There's a lot more mechanics involved because of the turtle's movement. So this is the first survey of loggerhead turtles using metabarcoding. And so they found that the differences in the community seem to be reflected based off of habitat as well as the anatomy of the host. There were different conditions, factors driving diversity, different conditions that played a role in microbiome composition. In the study included the origin, body site, turtle state, and also the sampling season. This is another important concept that we'll see with birds. And that was for bacterial communities, and the eukaryotic microbiota had a similar pattern. But there weren't as many differences between body sites. So an interesting curiosity that I came across was that the University of Central Florida has a marine turtle research group. I'm not going to play the video for you. It's on YouTube. It's just a couple of minutes long, but a fascinating look into a lab that is trying to study whether or not sea turtles can harness magnetotactic bacteria to basically get a sense of navigation. It's really interesting because it's been shown that animals will use magnetic fields, lots of different cues for knowing how to navigate with sea turtles. It's crucial as they're trying to find their way back so that they can nest. So how is this done? And the group at UCF is aware that there are metotactic bacteria, basically bacteria that can act as magnets. And so they developed a nice database, which you can access using the SRA database or sequence archive, I should say. And so this is a really interesting data set for those who are interested, where they've compiled different sequences related to bacteria that can act as these magnets. And this is under the efforts of Assistant Professor Fitak, who had a hypothesis that certain animals can use these bacteria as a means of navigation. So what he had his PhD student do is they collected tear ducts because these are connected to nerves, which could biologically have, this could be associated with a magnetic sense. And they found that turtles can produce a lot of goopy tears like every 30 seconds or so when they're on land to keep their eyes moist. And so they would collect these tear ducts or these tears from the tear ducts, and they were looking for bacteria in them. And they found evidence that these bacteria do reside in these animals, which is really cool. But how they're being used, if they're being used, has yet to be determined. So I'm interested to see where that goes, and I wanted to share that new bit of research with you. Now we'll talk about sharks. And remember, in comparison to the carapace of the turtles, there's something very unique about sharks. There are many different shark species, of course. So there are studies comparing different microbiota, commensal microbiota, based on the roles that the shark must play in the ecosystem, what environment they're navigating. For instance, bull sharks have to be able to swim through the rivers, so they're going to be potentially more vulnerable or more exposed, I should say, to pollution, to things along the river that you may or may not see in the ocean. And tiger sharks are notorious for eating a lot of things indiscriminately. So there is some interesting research comparing the microbiome of different shark species. There are shared OTUs in bony fish and cartilaginous fish like sharks, which suggest different roles in digestion and immunity. What's interesting is that while firmicutes and bacteroidetes dominate the terrestrial mammalian gut, proteobacteria are less common. Proteobacteria actually may dominate the fish gut microbiome. So this is quite interesting contrast. The gut microbiome of fish are defined as top piscivores, appears to be less diverse and less rich. Whereas an omnivorous shark species called the bonnethead is capable of breaking down seagrass, has fiber degradation capabilities, and that's possibly due to the bacteria that they have in their gut. Feeding studies also showed that there is a transient, so not the core microbiome, but a transient influence on the microbiome. This is seen in herbivorous and omnivorous fish species, which makes sense. And they're looking at chloroplast sequences for this. A couple of other interesting components are that teeth microbial communities can distinguish shark species. Pretty cool. And wounds are really crucial into understanding the microbiome of sharks. Wounds allow opportunistic bacteria to grow. However, this is typically only seen when dermal denticles are lost. And bacteria do differ by tissue sites. This is a common theme. Sharks have evolved mechanisms to protect themselves from pathogens, including mucus production and antimicrobial properties. Not something that we'll talk about in just a couple of moments. First, I want to present this nice study from 2015 where they did find that proteobacteria dominated the microbiomes of most species of sharks. There was not a single OTU shared among all species, although there were species in a core. There wasn't an OTU that was seen in all species, which is kind of interesting. As I mentioned, this was a study, very pivotal study that showed some specific differences in composition across different fish, including shark species. And they found that the lowest richness and diversity was seen in fish that were the top piscivores, so all sharks, barracuda, and mahi-mahi. And there was a photobacterium OTUs, collection of OTUs that dominated the core gut flora in the 3 shark species that they studied. And diet was one of the greatest drivers of richness and diversity, as we might expect. So this is a simple bar graph where we're looking at the percentage. So all of this is relative to one another, percentage of different components of the microbiome across these different species. And they did sample different individuals, so 3 different mahi, and we can see where there are some consistencies. The black sea bass, however, this one was definitely an outlier. Okay, so another study of sharks found that Maraxilacese was common to all 3 shark species that were studied, the blacktip, the bull, and the tiger. The blacktip had the highest diversity of cultured bacteria, and the healthy sites only cultured commensal bacteria. What was interesting was the wounded tissue. In the wounded tissue, they were able to culture pathogens like Vibrio, which is a significant pathogen in marine systems, and Macrococcus. Pseudo-Alteramonas has antimicrobial properties. And what was really interesting about that bacterium is that it was found in blacktip shark wounds and not in healthy tissues. These bacteria can digest the cell walls of Vibrio and lower infection risk. And it wasn't cultured from surrounding water, so this suggests that it's unique to the fish itself. So this could be a natural response of this particular shark to pathogens. Bull sharks, as I mentioned, they're quite unique for going up rivers and streams. These are Pseudoxanthomonas. They had this bacterium that can degrade organic pollutants and mercury. And what's kind of cool about that, as I mentioned, is if they're swimming up these rivers off the coast, then they're going to be exposed to a lot of things like this. They're more susceptible due to their movement into fresh water for periods of time. They didn't find any commensals in their wounded tissue. Then in the tiger sharks, they only find planococcus in the tiger sharks, and this was on their skin. This is a species also known for antibiotic compounds. So this was the first study to find it in a species of the Elasma branch. These sharks have protected mucoid layer, which could increase diversity, and Pseudo-Alteromonas was found on all tissue sites. So this was a very nice comparison of different species, and you can see how they've, it's a nice way of demonstrating where these families were located on each different species of shark, and then we see the Venn diagram showing where there's overlap. So where are we going with all of this? What's really interesting is there are people who have for quite some time been interested in harnessing the patterning on shark skin, these denticles, and what they found was that if they had antimicrobial resistant bacteria, and they were trying to see if they could grow these bacteria. They were trying to see if they could colonize, if they could attach when they were on these on a smooth layer. There was a great density of these microbes. However, when the team applied a micro pattern that mimicked the denticles, they found that there was far less growth. This was really significant. And given the rise of MRSA and MSSA, there's particular interest in this. Could we potentially take high touch areas in hospitals and mimic the scaly surface of a shark skin to try to curb antimicrobial resistance? This is a really cool paper showing that this micro pattern could have a lot of implications, not just for the health of the shark, but also for us in bioremediation and in protecting humanity. So, of course, there's a company that's working on developing this micro pattern. They have a nice website about their synthetic surface and what they're trying to do with it. Okay, now we'll shift gears and talk a little bit about bees. There are some nice studies on how the composition of the microbiome differs, mainly based on the behavior of the bee. So we're going to talk about that. Bee to bee interactions are pivotal in this landscape. Climate also has a role in pollen consumption. Composition also depends on the stage of life, and there seem to be interactions between bees that suggest horizontal microbial transfer. Thriving hives also have different microbiome capabilities. In particular, there are more bacteria that are associated with sugar degradation, which makes sense, right? Burkholderia are among the most prevalent microbes in bees. And there's also Bifidobacterium, Lactobacillus, which is really interesting. And auxiliary metabolic genes have been discovered in bacteriophage of honeybees in particular. So this is a study. I don't want to get, this is one of the better plots that they have, a heat map basically showing the average abundance as a number, and then prevalence. So the prevalence is how often this bacterium is seen in a particular area or in a particular species. So this is important so that we know how many individuals harness this microbe, and then in what average abundance. So I thought this was a nice way for them to demonstrate the composition across different types of bees: the honeybees, stingless bees, bumblebees, and then out groups. And we can appreciate from this heat map how different these two are, where they're mostly driven by these bacterium, whereas Bifidobacterium is highly prevalent in the honeybees, especially. And then there is a stingless bee where there's strong prevalence of Bifidobacterium and also a high abundance. So sociality in this study was found to play an important role in the microbiome. And I, again, just thought this was a really important way to show the results. And I thought you would appreciate that as we have been dissecting the literature all semester. So here the question was whether thriving and non-thriving honeybees could be separated based on bacterial composition at a community level. What was interesting was they did find higher diversity in the thriving communities, and that species that were higher in abundance are also those associated with sugar degradation. Non-thriving colonies in red across the board had much lower diversity, and they were depleted of certain health associated species. They had high abundance of Gilliamella apicola and pollen, which is disease associated, and a pollen degrading bacterium, Bifidobacterium asteroides. So much of the microbiome of the bumblebees appears to be interestingly transmitted vertically from the queen bee. And they also found that composition depends on size and life stage. And bumblebees actually possess less strain variation, and families are linked to polysaccharide digestion. There is a difference in the functional carbohydrate digestion capabilities between bumblebees and honeybees. Then in this study, they looked at different viral clusters. They were interested in knowing if the gut microbiome of honeybees was involved, and they were able to show basically the role of bacteriophages in the gut with respect to fermentation of pollen and nectar into honey. So they isolated from the hindguts different viral clusters, and they were able to identify a rich community of lytic and temperate phages across 118 different viral clusters. So the viral clusters are indicated with VCs and are connected, and we can see them being linked to lactobacillus, bifidobacterium, bartonella, etc. So now we're just going all around the world here. I want to spend a couple of moments talking about birds. Very diverse and complex. The composition will differ among birds, but function is quite similar. Proteobacteria, firmicutes, and Bacteroides are dominant members, and I'm sure you can guess at this point what some of the confounders are. But lifestyle plays an important role, sedentary versus migratory. And one of the key aspects I wanted to highlight for you guys in this lecture was the role of noise pollution. Yes, noise pollution in the microbiome. So not just dealing with the waste that our society is unfortunately generating, but also the noise that we're generating and how this can impact not just the health of the birds, but actually their microbiome as well. And the diversity of the feather microbiome appears to correlate with feather damaging bacteria, makes sense, and negatively with bacteriocin producing bacteria. So let's look at one example study where researchers were interested in Kirtland's warblers. They spend the winter in the Bahamas, where they have a very fruit-rich diet. And then they breed in Michigan, where their diet is driven by insects. So there's a change in habitat, of course, but then there's also this big change in their diet. And they found that in the Bahamas, the warblers were more characterized by firmicutes, which help degrade cellulose, and actinobacteria. But in Michigan, when they were breeding, they found that proteobacteria, which is often found in insectivorous species, was much higher. Cyanobacteria were present in the Bahamas, but then they were absent upon their return to Michigan. So this is just a typical box plot that we'll see with what's called jitter, where each of these dots is a different individual. And we see pairwise comparisons across the different sampling conditions. So the initial capture in the Bahamas, the recapture, and then another recapture, and how different the composition is between the Bahamas and Michigan with proteobacteria and cyanobacteria and actinobacteria, et cetera. So, with respect to noise pollution, this can lead to a lot of changes in stress hormones and foraging behaviors, as you can imagine in the wild. But the gut microbiome can also be affected by this. So, noise was found to increase sterone and decrease food intake in sparrows. And as you can imagine, this has pretty important consequences on the microbiome too. So, birds were found to have a higher alpha diversity during noise exposure. And what's interesting to me is this could help explain differences in bacterial richness when we compare urban and rural areas. And this has some similarity in theme to what we've talked about with industrialized societies and looking at like the microbiome, for instance, of the Hadsta infants. So just thinking about how urbanization, industrialization, pollution, like different themes can have a really important impact on the ecosystem, and in some ways, very, very, very small things like the microbiome that can have large consequences. So it wouldn't be fair to talk about birds and not talk about crocodiles, especially since we're at the University of Florida. So this was actually a really beautiful schematic that was developed by former students of the microbiome course. So I thought that they did a very clever job of showing how composition differs based on body site. And again, this is a consistent theme that we see where firmicutes is the dominating bacterium in the stomach, duodenum, the jejunum, and less so in the ileum, where fusobacteria are more dominant. So this is a nice reminder of how different the body sites can be. So Jurgena Stanley was also very kind to speak with our former students. And I just thought she had a very interesting story, and I wanted to share that with you. She was actually raised on a farm as a child, so she'd been around animals and had a great interest in them. She there was some talk about veterinary school, of course. But that was not a common career to pursue in her hometown. So she took genetics and microbiology and became very interested in the animal microbiome. And so that's something I wanted to share with you. Just an interest that started in childhood, leading into a wonderful research career. She immigrated to Australia because in Yugoslavia there was war in her country, and now she's a professor at Central Queensland University School of Medical and Applied Sciences. She has a PhD in molecular microbiology and studies bioinformatics, immunology, environmental microbiology. And she did note with the students that one of the most difficult things can be securing the right funding. So getting a good team together to work efficiently and to get to overcome funding limitations. And she had said that she was felt very lucky to be able to acquire the funding that she needed to look more into these fascinating creatures. She's been published in a lot of different high profile journals, and she's worked on other animals, including livestock. So I thought that was a nice opportunity to just share with you what former students have learned and how they were able to use this class to connect with somebody outside of the university in this field. And she was very gracious with her time. So what this group has found is that interestingly, crocodiles, their gut is composed of two very primary pathogens, or what we think of as pathogens: Cetobacterium someri and Clostridium difficile. So that's fairly interesting. They can have a very high abundance of Clostridium difficile that's commonly pathogenic in mammals and in birds, but it exists without pathogenesis in some species of crocodiles. So that's quite interesting that they can dominate these animals. Stressors that can stunt young crocodile growth also increase risk of infection. And it's thought that the tolerance of pathogenic bacteria could be associated with their diet. Um, there, so it was very interesting. And this group was looking at farmed saltwater crocodiles, again, to find the most abundant microbes in different sections of the digestive tract. And no research had previously investigated this with respect to production efficiency. Dinosaurs. So, I suppose I was interested in at least having a slide or two on this because I found this quite curious, and the connection to birds. But I came across this a while back that fossil bones could potentially, again, this may be speculative, but could be a host to a microbiome that is distinct from surrounding sediment, just as we saw with the sharks. They had these species that were distinct from the surrounding water. And some nucleic acid analysis has been done, revealing a diverse community within dinosaur bone, which suggests it could be biologically active, right? There's also discussion among experts on whether dinosaurs benefited from social interactions for commensal bacteria transfer, and wondering if horizontal transfer of symbiotic bacteria was a driver for socialization. So here is a study looking at organic remains, finding that the remains may persist in fossil bones depending on different biomolecular conditions. And they've taken exogenous organic material, has been found, like amino acids, but no evidence of ancient endogenous proteins. Fossil bone could be a nutrient dense habitat for soil, for moisture retention, and could be biologically active. It's uncertain whether the microenvironment creates anaerobic niches for metabolism. But they were looking at especially the abundance of delta proteobacteria in these samples, which could help explain pyrite and iron oxidation in fossil bone. So again, just a really interesting study of the bone core of a Centrosaurus, and then the bone core here after they demineralized, a subterranean bone, and then the scrapings around the surface from the subterranean bone, and the different bacteria that they were able to recover based off of this. So for hyenas, I thought that we should have a little bit of representation from some amazing wildlife in Africa, and for this, I want you to watch a short clip, not this entire webinar, but just the clip here from Connie Rojas at Michigan State University. And she lays out just a nice, simple sampling study that they did out of Kenya on juvenile and adult hyenas. 13 females, 11 males for the juveniles, and 12 female adults. And they applied 16S rRNA gene sequencing to these samples, and were able to study different factors that could predict the microbiome. Not surprisingly, diet, habitat, social relatedness across 6 different body sites. And they looked into body site specificity and how the structure varied by host behavior. So I just want you to be familiar with how these webinars go. So I'm linking that in the module, and you can watch that as part of your required videos. And now on to the primates. So let's talk about these primates. Social networks strongly predict the structure of the microbiome and the genes that are encoded. So physical contact is a very strong determinant of composition and can have great consequences on health. An age-related microbiome variation could actually just be due to diet or lifestyle, not necessarily the age of the animal itself. So here we have another way of representing the bacteria, but instead we have fold enrichment. So this is capturing how different the bacteria are based on their comparison. So first between social groups here, and then in B, they were looking at a specific grooming network. And then here between and within groups at the species and genus level. What they found was social group membership was the single strongest predictor of microbiome composition and could explain almost 19%, 18.6% of global variation, and 10.8% of enzyme ortholog variants. Grooming networks were also pretty important. The association with social membership remained even after controlling for genetic relatedness, which I thought was really cool. Differences were not likely to be explained by diet because only one diet component differed between the two groups, right? So if we pretty much have a common diet between the two groups, and that can't be what's driving the differences, right? So there are ways that we can kind of apply that same methodology in other cohorts, like doing propensity score matching, where we ensure that our controls, we have the same representation of certain factors that could drive composition in the controls and in the cases. And if we still see an effect, that means it can't necessarily be due to that factor because there are no differences between cases and controls in that. There were also no differences in fruit associated bacterial enzymes. So that's another indicator that they had that it wasn't driven by diet. The socially structured bacteria were anaerobic and non-spore forming, and this was the first evidence in vertebrates that social effects extend to composition, which could also give pretty great implications for social interactions in health. So to follow, to finish up this lecture, where we explored all these different, some curiosities from all these different species across the globe, I just wanted to end with a few more concepts that temperature does impact animal fitness, of course. But the microbiome also responds to temperature, which suggests some microbiome plasticity. Extreme temperatures can be linked sometimes causally to effects on the host, but the gut microbiome can also impact host fitness, colonization resistance, energy nutrient assimilation, fecundity, development time, etc. Changes due to changing season are thought to potentially reflect diet changes, but could also be due to changes in temperature as well. So studies of the microbiome in wild living hosts is necessary, just like we saw with the warblers. Very few studies demonstrate a causal role, but that would help a lot in predicting responses to things like climate change. I did want to give an example that in Gelatis, Ethiopia, it was found that metabolic flexibility via the microbiome allows for specialized diets and maintenance of energy during thermoregulatory stress and reduced food availability. So what they found was bacterial genes in amino acid and lipid metabolism and energy were increased during cold and dry periods. And then microbial changes were driven by seasons. So number one, cellulolytic fermentative bacteria that digest grass were seen in the rainy seasons. And then versus number two, bacteria that break down starch in underground plants was seen in dry seasons. So that was a pretty cool little study that just, again, recapitulates that temperature can have an important effect. You are going to read this paper. It's a very nice perspective on social interaction, since that seems to be the driving force for this module in this area of research. Social interactions provide opportunities for what's known as the social microbiome. The core aspect is belonging to a certain social group, and it's not just species specific or an entire population. Proximity and connection decrease beta diversity. And they describe these archipelagos as a symbol for individual host microbiomes within a social network. Different levels in the paper include individual, within group, between group, population species, and sympatric species. And there are known examples of each, which the paper will go over. And finally, microbial transmission is not limited to pathogens. That should be a big takeaway for you from this. Commensal and mutualistic gut microbes are transferred through social contact. The social microbiome can have very big implications for fitness and animal physiology. And finally, I thought this was very cool studies looking into captivity versus animals in the wild. So there was a nice study, McKenzie 2017, which is also in the module, where they looked at paired wild and captive samples from mammalian taxa at 6 different orders represented here. And they were investigating generalizable patterns of captivity on the gut microbiome. So they would compare them to their wild counterparts, and they found that alpha diversity remained the same in some mammalian hosts like giraffes and anteaters and aardvarks and bovids. There were declines in captive conditions in some hosts like canids, primates, and equids, and increases in the captive condition, but only in one host taxon, and that was the rhinoceros. This has really important implications for thinking about biodiversity and breeding strategies. But the differences in beta diversity between captive and wild state were also observed for most of the taxa, except for bovids and giraffes. It was influenced by host taxonomic group, diet, and gut fermentation physiology, especially. So the larger shifts in relative abundance were firmicutes and bacteroidetes. And building off of that, one more study showing that captivity of certain primate species resulted in the loss of different native microbes and colonization of Prevotella and Bacteroides, which actually dominates the human gut microbiome. So that's kind of interesting. So the primates were converging towards a human microbiome in that case. And this captivity parallels modernization that we see in humans, again, thinking back to like the Hadsta study. So these are, again, just a few curiosities that I wanted to share with you in this really cool module. And now I would like to know what you're curious about. I purposely didn't talk about dogs, and that's because there's a lot of really cool literature out there, and I imagine many of you would want to cover that. So I didn't want to steal that opportunity from you necessarily. So here are a few very interesting curiosities that I wanted to share with you. And as part of your exercise this week, I want you to look in the literature, look in the news, and try to find something that speaks to you and share it with the class. Well, thank you so much again. I hope that you enjoyed this module, which is a bit different from others. And I look forward to hearing what you have to share. As I started at the beginning of this lecture, the best way to inspire change in our planet and protecting one another is educating ourselves and allowing ourselves the opportunity to connect with a specific animal and realizing how remarkable other life forms can be. I don't want us to think that we're studying the microbiome of animals purely for human benefit, but just because they're wonderful to study in their own right. So ecologically, culturally, personally, biodiversity is so important. And I hope that this module has inspired some of you. Thanks and have a great day.